Okay, so we're going to do tissues first. Okay, so let's take a look at the tissues. And because of, we'll be lecturing also in lab as well, so just FYI type of thing. All right, so with tissues here, we're going to first learn some terms. Histology. When we look at histology, this is the study of tissues. So when we look at tissues or we're studying it, then that's what histology is. What is tissues? Tissue is a set of cells that perform a function. Okay, and we're going to be learning that there's several different types of tissues. There's 4 main types of tissues, but they also have this extracellular matrix involved as well. And what the extracellular matrix is things that are outside of the cell. Okay, so the neighboring area outside of the cell. It's going to consist of some sort of liquid, if it has a liquid extracellular matrix. Some of them has a solid extracellular matrix, and we'll talk about that later. But also it contains these fibers as well as other macromolecules. Macromolecules means big molecules. Macro means big molecules. 4 main types of tissues. We have epithelial tissues, which usually covers things and protects things. We also have connective tissue, like the name implies. It's going to connect tissues with each other. We have muscle tissue, and when we talk about muscle tissues, this is helping with contraction. So it makes things so that we can provide some sort of movement. And then lastly, we have nervous tissue. And nervous tissue will help with communication purposes. Okay, so let's first look at epithelial tissue, right? We have 4 different types of tissue. We're looking just at epithelial tissue for right now. So this covers and lines the surface of our organs, and it also makes up our glands as well. And we'll talk about the glands later on. Here are some of the characteristics. First of all, there is little to no extracellular matrix. Remember, extracellular matrix is the stuff outside of the cell. So there's not going to be that much stuff outside of the cell. So when you look under the microscope, you're going to see a lot of cells and not so much space in between those cells. And then there will be different surfaces as well on these epithelial tissue cells. Okay, so when we look at a cell, the apical surface here is going to be the top surface. So if you take a look at the cell here in this drawing, the apical surface is on the top here. The apical surface is usually towards the outside environment or to this space. And when we talk about space within an organ, there's a term for that. This space that's within an organ is called the lumen. Okay, it's called lumen, L-U-M-E-N. And then there's these lateral surfaces. The lateral surfaces are going to be the side surface of these cells. So right here on the sides here are going to be the lateral surface. And then on the bottom is going to be the basal surface, which makes sense, right? It's on the base. And this basal surface is going to be connected to connective tissue. And there's like a little structure right in between the connective tissue and epithelial tissue called the basement membrane. So this basement membrane connects the 2 epithelial tissue and connective tissue together. The reason why we want to have this connection is because epithelial tissue is avascular. Okay? When I say avascular, it means that it has no blood vessels. Avascular. Vascular means blood vessels or containing blood vessels. And this A means no, or the opposite, basically. So it has no blood vessels here. If it doesn't have blood vessels, it's not going to get nutrients. It's not going to be able to get oxygen and carbon dioxide and dispose of its waste and carbon dioxide. So it needs to rely on the connective tissue that's right next to it in order to get these nutrients and dispose of its waste. Okay. So Later on, when we take a look at the different types of membranes, most of the membranes will have both epithelial tissue as well as connective tissue with it. As for, we will be dividing up the groups into labs once you go into the lab. For the lecture, it is everyone has to join in the same lecture. I don't think if I do it as breakout groups, then you won't be able to hear me lecturing in this. It doesn't have that function that I know of. Once again, the apical surface is on the surface, right? It's next to the top here. Okay, so because it's close to the environment, it's going to be damaged. And because if it gets damaged, then we need something to replace it. How do we replace it? Well, we replace it by having cells undergo mitosis. And mitosis is cell division, so it will end up replacing the damaged cells that are above it. And this usually occurs using stem cells. So that's some characteristics here. There is one more thing down here. I can't seem to see it, but let's see. What does that say? Give me a second. Just, that's functions. Okay, yeah, I already mentioned about the lumen, right? It's that space within hollow organs. I mentioned it up here when we talked about how it covers the part that's the outside environment or some body space, right, kind of thing that we just talked about. All right, so let's go into functions now for these epithelial tissue. Since it covers things, it serves as a protective barrier, right? So regarding to your phone numbers, just send it when we go into our labs. Don't do it right now. We could just go into our labs and we'll send it there. Okay. So with protection, it protects our surface. Abrasion is when you end up rubbing against something. So if you rub against something, the epithelial tissue will try to protect it by prevent. Even if you cause abrasion, it will just take off the surface layers of it and still have some layers underneath it. Dehydration prevents dehydration. We'll talk more about that when we get to the integumentary system, and also damage as well. It is selectively permeable. When I say selectively permeable, it means only certain types of things can be permeable. Permeable means going through. Okay. So it depends on what it is. It allows certain things to pass through and other things to prevent it from being passed through. And then we have secretions. Okay? And secretions are when something is being produced and is getting released out into the environment. So some of these epithelial tissue will have these secretions, and we'll talk more about this like glands. And then we have absorption. Absorption is like the reverse of secretions. Instead of releasing something, it brings things into the cell. And then we have filtration as well. And when we talk about filtration, now we're talking about something that kind of like a coffee filter. So it has to move through a sieve-like membrane. So these molecules will end up moving through some of the epithelial tissue. When we talk about the urinary system, we'll talk more about this. But it will go through these cells and get filtered through like that. And then there's sensation as well. So those that are usually, depending on what we're talking about, but some epithelial tissue will have nerve endings to it, and it will detect different types of senses. Usually some sort of general sense. Okay, so those are these functions here. Okay, So before we go into more about the epithelial tissue, I want to look at these cell junctions. And these cell junctions are how these cells within the epithelial tissue gets connected to each other. And it's done at the lateral surface. Remember, the lateral surface is on either sides over here. There's tight junctions, and tight junctions we'll talk about, but basically they're so tight that prevents things from passing through these cells or between the cells. Don't worry about adhering junctions. Desmosomes, so here are the tight junctions over here. The desmosomes we'll talk about later, but they're kind of like a Velcro kind of thing, keeping the neighboring cells together. And also gap junctions. And gap junctions, like the name implies, there's little gaps to allow communication between the cells. So let's look at the tight junctions first. Okay. The tight junctions, it's closer to the apical side. What does apical mean again? It's on what surface? Is it on the top, on the side? Yeah, it's on the top. Very good. Okay. So sometimes I'll ask these questions, and while I'm asking this question, please type it into chat, and then just so that I could see that you're following along. Okay. Yeah, thank you. And then since these are tight, they will end up preventing things from passing between the cells, which makes sense too. The reason why we want to have this is because, say, our stomach, right? Our stomach has stomach acids inside of it. Do we want the stomach acids to go through the cell or between the cells to damage our deeper structures in our stomach? No, right? We don't want that to occur. So we want to prevent the acids from passing through by having these tight junctions here. So the GI tract.
Professor, I'm sorry. Do you mind slowing down just a little bit?
Yes. Okay. Is there something that you're missing, or?
No, it's just you talk really fast, and I'm taking notes. So that's all.
Okay. Just let me know. So GI, what that means is gastrointestinal.
What's LE?
LE?
Yeah.
Where do you see LE?
Right before GL track, there's like LE.
Oh,
I think it's
example. That means example.
Oh, okay.
No
Thanks.
worries. Yeah. G, gastro means the stomach. Intestinal, the intestines. And the intestines will have a lot of, small intestine especially when we're talking about it, it has a lot of enzymes. Desmosomes, on the other hand, they are located here on the lateral surface as well, and also sometimes on the basal surface. It's not shown on this diagram, but I'll talk about that in a sec. Okay, so this is using intermediate and protein filaments. So there's proteins once again, and it becomes more like a button. Okay. Sometimes it's a half desmosome, and that's the one on the base. So this is to prevent mechanical stress. So it prevents them from being torn apart from each other. Okay, so it keeps them together. And then there's hemidesmosome. Since this is being recorded, you could always go back and listen to it again and finish up the parts that you didn't finish up. We have a lot to cover at the same time, so that's why I'm trying to keep up pace as well. But I will try to slow down a little bit, but I do tend to talk a little bit fast. So like I said, there are hemidesmosomes as well. Hemi, it's kind of like semi. Like when we talk about, so this means not totally, it's like half. So this is a half desmosome, like the one that I was telling you about is on the base here. And this hemidesmosome is to prevent the epithelial layer from being torn away from the connective tissue. Okay, so it helps anchor it on the basement membrane. So once again, this is to prevent mechanical from being ripped apart from each other. Semi or half. Decimus. Alright. Just make sure. Okay. So one thing I think I forgot to mention, for tight junctions, they use adhering proteins. For this desmosomes, they use intermediate and protein filaments. They're all proteins, but they're slightly different from each other. And for gap junctions, they use proteins called connexon. Okay. So gap junctions, like the name implies, has little gaps in between. And these gaps allow them to communicate with each other. So they tried to depict it over here. So they have these little gaps so that they could communicate with each other. This is very important when we start talking about the cardiac muscle. Cardiac muscle is your heart, right? So that way you could have your heart contract all at the same time and not have one part of the heart contract first and then another part of the heart contracting later. You want it all contracting at the same time, so it'll be most efficient on pumping blood. So by doing this, you end up, just say the cell gets excited. When this cell gets excited, it's sending these molecules over to get this cell excited, which then sends molecules over to get this cell excited. That way, when it contracts, all of them will end up contracting at the same time. You also find this in the respiratory tract as well. Cilia are these little structures that move the mucus towards their pharynx to your throat. All right, let's go ahead and see if we could do this real quick. Let me set this up, though. I did not set it up prior to it. But let's see if you could go to this website here. If you end up having a QR reader, you could use the QR reader as well.
Do we just click on the link on the slide, or do we copy and paste it somewhere?
What was that?
Do we, like, how do we select the link? Do I type it in?
Oh yeah, let me link it to you. Actually, that's good thing you mentioned that. I'll put it in chat so you could just click on it.
Oh, okay. Oh, okay. Thank you.
There you go. Thanks for saying something about it. And once everyone's in, okay.
Got a room name? It's still room name.
While you're going
in. It's asking for a student login.
You don't need to log in. If you click on this link here, it should just ask you for a room name.
I just clicked on it and it's saying it needs a login.
Oh, interesting.
Did you click the link in the chat by any chance?
Yeah.
Oh.
That's odd. Don't worry. Well, I guess in that case, you don't have to actually log in and stuff. Just answer it on your own. Someone already answered the question. But anyways, if you're not in here, that's okay. Just try to answer this next question here. Okay.
Is it normal for no questions to be on the screen at all? Like
this is the question I'm going to ask, and this is where you're going to type. Select A, B, or C, or D, or E here. So go ahead and try to answer this and see. Pause the video at what you selected. Okay. Most of you ended up choosing A, which is the correct answer. So when we talk about these cell adhesions, whether it's the desmosomes, the tight junction, or gap junction, we're talking about epithelial tissue. Connective tissue won't have these particular proteins and stuff that will connect the cells together. They have more of an extracellular matrix that we kind of talked about. Sometimes they will have it, but not all of them will have these gap junctions, or sorry, these cell adhesion. We talked about earlier cardiac muscle. They have it, but not all of them will have the desmosomes as well as the tight junctions as well. So A will be more of the correct answer, although there are other tissues that might have certain aspects of it. Good. All right, so we're still talking about epithelial tissue, but now we're going to be talking about glands regarding to epithelial tissue. Okay, so glands are going to be, once again, from the epithelial tissue, but sometimes they could be just one single cell. They have a secretory function. Remember, secretions means that it's going to end up releasing something out to the environment. So it's going to be, in this case scenario, it's going to be whether it's sweat or whether it's mucus, or sorry, mucin, which becomes mucus, so on and so forth. Okay. So hormones as well, we will talk about the endocrine glands are going to be releasing hormones, like we just said, enzymes, mucin, so on and so forth. So there are the endocrine glands. The endocrine glands are going to be ductless glands, meaning that it doesn't have a duct. And these are going to be secreting hormones. Okay. And when they release this hormone, it's going to release it into what we call the interstitial fluid, which is the tissue fluid. And then it goes into the bloodstream. And where does the blood go? It goes everywhere into the body, pretty much. And then there's these exocrine glands. So the difference between the two is that the exocrine glands has ducts. And the ducts will release it to the surface. And once it releases to the surface, then it could evaporate if we're talking about sweat to cool us down. It could release enzymes, which helps degrade proteins and other types of molecules, whether there's carbohydrates, so on and so forth. Saliva is an example of that as well. It could be categorized by the number of cells and also how it's secreted. And we'll go into those details in a second. Okay, so this is kind of like the overview. This is what you have over here, but I'm going to go into each one of these. So just hang on there, okay? Right. So first of all, we could categorize it by the number of cells. So depending on how many cells there are, if it's unicellular, uni over here means 1. Okay. So it means that if it's only one cell, it may or may not have a duct. But we talked about this earlier. Exocrine glands tends to have ducts. Okay. This is the only exception. It may not have a duct if it's unicellular. And the one example that we'll be talking about is the goblet cell. In goblet cells, it's named to look like a goblet, like a cup. And you can see it from this histological picture here. Take a look at it. This is a goblet. Okay, it has this kind of shape to it. Okay, and it's going to release mucin. And when mucin mixes up with water, it becomes mucus. Marlene, you can't see all the notes on the screen. Can you help? What do you mean you can't see? Oh, I see what you're talking about. Yeah, or if you double click on the screen, it will reduce it as well. And then there's multicellular exocrine glands as well. Okay, multicellular means that multi means many. So there's more than one. And they end up containing what we call acini. And acini are these sacs that's going to produce these secretions into the ducts. And examples of these are like the salivary glands, and also our pancreas, and so many other glands as well. Okay. So, okay, and then there's secretion method, right? The previous slide we were talking about the number of cells that there are. And this next one here is how they secrete things. Okay, so we have merocrine glands, and when they secrete it, they package these secretion in vesicles. For those of you who don't know what a vesicle is, it's kind of like a membrane-bound bubble. Just think of it like a bubble within the cell. So there's the secretions inside these bubbles, and then they get released into the environment. Examples of this is the saliva, tears, and sweats, and we'll talk about the eccrine glands later on in this particular unit. And also most of our enzymes from our gastrointestinal tract will be using these merocrine glands secretions. And then there's these apocrine glands. The apocrine glands are slightly different. They still release secretions, but they release it at the apical portion of the cell. What does apical mean again? It means
The cell. The top.
Yeah, the top. Yeah,
the top surface.
Very good. Okay, and they're going to pinch off from the top and release it. An example of these are the axillary region. Axillary is your armpit, and we'll talk about that in a second. Inguinal region is your groin area. Anal region, hopefully that's a familiar term around the anus. And also the mammary glands. Okay. So usually these over here are going to be releasing like pheromones kind of thing. Then there's the holocrine glands. And the holocrine glands, they're more thicker. They're more viscous. When we talk about viscous, it's more thicker. That's what it means here. So this is going to be thicker. Kind of like honey, you know, or molasses. They're really thick. The reason why it's thicker is because the cell will disintegrate. And when the cell disintegrates, the cell's outer part of the cell that surrounds it, does anybody know what that structure is called that surrounds the cell? Plasma. Yeah, plasma what? Plasma membrane. Yes, very good. It's a plasma membrane. And the plasma membrane is going to be made of these structures called phospholipids. There's the lipid portion of it. And since it's a lipid, it's more fatty-like. And so it's going to be more viscous because of that. An example of this are the sebaceous glands. And the sebaceous glands are going to be found on our skin. And if the duct of the sebaceous glands gets covered or gets blocked, it could potentially cause acne. Acne is pimples, basically. All right. Before we continue on with epithelial cell layers, I want to make sure that we have enough time to do this next part here. Okay, so those of you who are enrolled in the course, I want you to go onto Canvas here. And you're going to scroll all the way down to this Lecture 1 in-class assignment. Okay, so if you're already on the page, you might need to refresh it before you do it. Okay, so everyone take a little bit of time to actually do this. For those of you who are on the wait list, what I'm going to do is I'm going to send you a link here for you to fill out. So give me a second for those of you who are on the wait list. Okay, so remember, those of you who are on the wait list will be clicking on this link that I'm going to be putting onto the chat. Those of you who are enrolled, do not click on this link, okay? Since you'll be clicking on the in-class assignment here. If you're already enrolled in class, click on this lecture 1. For those of you who are on the wait list, click on this link over here. Visible body, unfortunately, has not been fixed. I'm still dealing with the representative with visible body, but should be free. So don't purchase visible body. So it's not. So remember, those of you who are on the wait list, please click on this link here. Those of you who are enrolled, go to the Canvas site and go to lecture 1 here.
Wait, what is visible body work?
Visible body is kind of like this 3D model kind of thing where you could rotate it around. It should be, it's only an optional thing. You don't have to use it. It's more like a resource for you to use. So you don't have to have it.
Professor, what email do we input?
I guess whatever you want to whatever you use more frequently and check more frequently.
Professor, I'm currently enrolled in the class and I went to Canvas and in-class assignments, but there says that there's no assignments in there.
Yeah, I can't see.
Yeah, mine's too.
Refresh the page and see.
I did. I refreshed it and closed Canvas 3 times, but it's still not there.
No lecture pops up.
It's all the way at the bottom.
It's on the very bottom. Yeah, you gotta scroll all
It's-
the way at the bottom.
It's
I'm
different
sorry,
from
it just
his.
came up.
It looks different from his, Professor. That's why.
Oh
Yeah, it does.
Because I have all these other units here, right?
Yeah. Well, I mean, there's only one. I don't know. It just looks different.
Yeah, there's only one over here. Yeah, there's only one. This other one I haven't released yet.
There was just one question.
Yes.
Oh, I see it.
Where do we find the visible body work if we want to see if it works for me?
Okay. If you want to see visible body, it's over here. If you click
Oh.
on it, it should allow you to see something like this, okay? But there's a lot of people who are saying that they don't see this. But the reason why I bring this up is you could use this. Remember, these are all optional. You don't have to use any of this. But if you want extra practice, you could go through these assignments here. But the main reason why I want you to use this is optional once again. You go to My Apps. Once you go to My Apps, click on this Anatomy and Physiology icon. And then it takes a long time for this to load, but eventually it will load up. And once it loads up, now you could actually take the different organs and be able to rotate them around and whatnot type of thing. Once again, it takes a while for it to load, but eventually it'll load up. And then just say you go into the skeletal system and go, okay, I want to take a look at flat bones of the skull. Okay, and then you could just take one of these, you could kind of rotate it around, whatnot, zoom in, zoom out, so on and so forth.
We have lab exams, or is this just for extra practice, or do we use that other link that you said for lab?
This is optional. The APR, the Anatomy Physiology Reveal, that's for dissection purposes, and that's why I want you to have it so that you could look at pictures of the cadaver.
Okay.
But I haven't released that out because we have issues with that as well. So all these online technical issues that we've been having recently. But I'll let you know when it's up, though.
Sounds good.
Okay. For those of you who are in lab, we'll reconvene at 11:10. Those of you who have Tuesdays and Thursdays lab, I'll see you either in lab or on Wednesday's lecture. I do have office hours today, so if you want to come during office hours, I might be dealing with a lot of adding during that time. So maybe come towards the end of office hours if you have any questions and whatnot.
Professor, I have a question.
Yes.